Hello everyone, I am Rudula and today's topic is the biomarkers. Biomarkers are also called as the biological markers. A biological marker is a measurable indicator that can tell us something about person's health or disease state. For example, disease or pathological process in the body. For example, disease stage. Then biological process in the body. For example, heartbeat, blood pressure, and the temperature. Then a person's response to a treatment or medicine. Then or a psychological condition. Biomarkers are used in many scientific fields in different ways at different stages of medicine's development. The accuracy of biomarkers can vary, therefore, not all biomarkers are suitable for medicine development. Examples of biomarkers: the glucose level that are used as biomarkers in managing diabetes. Then brain images such as magnetic resonance imaging, that is MRI, that can provide information about the progression of multiple sclerosis. Then biological substances such as enzymes, which may be found in the blood or tissue samples and are often used in the cancer research. Then genetic changes and medical images or the x-rays. The aim of the biomarker use is to improve the process of medicinal development. Then clinical trials seek to measure patients' response to a treatment. Then it is not possible to measure the response directly. Biomarkers may provide an alternative way of measuring an outcome. They serve as surrogate endpoints. Then these are the advantages of using validated biomarkers as surrogate endpoints. They might be able to be measured earlier, then more easily or frequently with the high precision. Then they may be less affected by other treatments, then reduce the sample size required and allow research to make faster decisions. Then they often important ethical advantages as surrogate endpoints in disease with poor uh, prognosis. Then the example of the use of biomarkers as the surrogate endpoint. An example of the use of biomarker as a surrogate endpoint comes from the development of an antiretroviral medicine for HIV and AIDS. Previously studied would have been based on hard clinical endpoints such as the progression of the HIV infection to AIDS or patient survival. Then now cell changes such as level of CD4 lymphocytes, then and changes in the level of HIV virus RNA in plasma can be used as the surrogate endpoints. Aims of the biomarker use are second aim is to tailoring treatment to individuals. Biomarkers research is helping to improve how well we can predict a person's risk of disease. Then how a disease might progress once it is diagnosed. Then how an individual will respond to a medicine. This will enable safer and more effective treatment decisions. Then biomarkers in medicine development. Cancer, that is oncology research, was one of the first areas where the use of biomarkers was adopted. Biomarkers are used to make exploratory trials and phase second trials, that is proof of concept trials of medicines, more efficient. Only a limited number of biomarkers can be used for clinical endpoints in phase second trials, that is confirmatory trials. Biomarkers may be used in late stage trials in combination with the clinical outcomes, that is the clinical endpoints. For some medicines, only a minority of patients might respond. Because it is helpful to identify these patients for clinical trials using the biomarkers. The companion diagnostics are the tests that are validated and approved for marketing alongside a new medicine. These tests may help to select patients likely to respond to a medicine, then exclude those patients likely to have an adverse reaction, and determine the best dose for a patient. Then many companies developing target therapies for cancer have also begun to consider the potential benefits of developing a diagnostic to pair with that treatment. The trend to develop medicines and companion diagnostics together rather than have both development happen separately. Now the biomarkers potential to increase the efficiency of medicine development. speeding up clinical trials. Biomarkers can be used to detect an effect or a lack of effect earlier and more frequently than if only a clinical outcome, that is endpoint is used. For example, uh, a panel of biomarker has been used in the early phases of a clinical trial for a psoriasis treatment. The biomarkers included epidermal thickness, that is the thickness of the outer layer of the skin, and the activity of several genes. These were both measured in the tissue samples. Then the streamlining clinical trials. Biomarkers are used to identify those patients who are more suitable for a treatment. Specifically, genomic biomarkers can be used to identify patients with a particular disease subtype or diversity or severity, then exclude patients at increased risk of serious side effects, that is adverse reaction. For example, melanoma patients are at risk of their condition getting worse if their tumors do not have a certain mutation in the BRAF gene and they are treated with kinase inhibitors. Identify patients with a high chance of benefiting from a particular medicine. Then improve our understanding. Biomarkers can improve understanding of how new medicines work and may lead to the novel approaches to medicines development in both non-clinical and the clinical phases. Then improving the ethical of trial recruitment. Biomarkers can help exclude people who won't benefit from starting non-helpful treatment, thus providing an ethical benefit. Then improving trial monitoring and stopping unhelpful trial early. Biomarkers may help decide whether to stop a trial early if there is no benefit to be gained by the patients in the trial, or an obvious benefit gained where withholding treatment would be unethical. Then the speeding up authorization. A medicine that is having a positive effect may be authorized soon based on information provided using biomarkers, and therefore may be prescribed earlier to a patient who will benefit. Then the challenges of using biomarkers in medicine development are the as the use of biomarkers in pharmaceutical research grows, companies face new technical challenge, then regulatory challenge and the ethical challenge.